Hello everyone. Today we are going to read about another topic in the glycolysis. Uh, Today we are going to read about two effects. One is Pasteur effect and another is Crabtree effect. First of all, let us discuss about the Pasteur effect. The Pasteur effect is the inhibition of the glycolysis by the oxygen. So basically the inhibition of the glycolysis by the aerobic condition is known as Pasteur effect. In the Pasteur effect, what happened in the aerobic condition? In the presence of the oxygen, the levels of the glycolytic intermediates from the fructose 1,6 bisphosphate onwards decreases, while the earlier intermediates accumulates. This clearly indicates that Pasteur effect is due to the inhibition of the enzyme phosphofructokinase. The inhibitory effect of the citrate ATP produced in the presence of oxygen on the phosphofructokinase explain the Pasteur effect. Now let us discuss about the Crabtree effect. Crabtree effect is just the opposite to that of the Pasteur effect. The Crabtree effect is the inhibition of the oxygen consumption by the addition of the glucose to the tissue. So basically the phenomenon of the inhibition of oxygen consumption by the addition of the glucose to tissue having high aerobic glycolysis is known as Crabtree effect. As I have already told, it is basically opposite to that of Pasteur effect. The Crabtree effect is due to the increased competition of the glycolysis for inorganic phosphate and the NAD+. So again I am going to tell you. The Crabtree effect is due to the increased competition of the glycolysis for inorganic phosphate and NAD+, which limits their availability for the phosphorylation and oxidation. Now let us discuss about the glycolysis and dental caries. Mainly the refined and the fermentable sugar basically inputs the dental caries in our teeth. The dental caries refers to the destruction or decalcification of the hard teeth due to organic acid released by the bacterial infection. The anaerobic bacteria, for example, Streptococcus mutans and Lactobacillus, that colonize the oral cavity contribute the development of dental caries. So these bacteria, anaerobic bacteria, grows optimally on the refined and the fermentable sugar, for example, sucrose of chocolates and candies, by utilizing the anaerobic glycolysis. Lactic acid and other acids produced by the bacteria erode the tooth enamel and dentin and dissolves the hydroxyapatite matrix of the teeth that result in the cavity formation. The low level of fluoride from the toothpaste when applied topically or when doing brush inhibits the enzyme enolase, which reduces the glycolysis and the tooth decay is reduced. So further fluoride integrates into the tooth surface to form the fluoroapatite, which offers the resistance to the demineralization. So you have to know that. In the topic glycolysis, I have told you the effect of the fluoride that inhibits the progression of the glycolysis in the RBC. So the fluoride especially inhibits the enzyme enolase, which will decrease the reaction, which will decrease the cycle glycolysis, which will inhibit the effect of the tooth decay, which will inhibit the effect of the bacteria in our teeth. So these are the brief explanation of the Pasteur effect, Crabtree effect, and the glycolysis and dental caries. I hope you understood this lecture. Thank you.